Recording on now. So welcome to the Soil CRC for the West Community of Practice online web workshop. We're going to be looking at soil constraints, and I'd like to welcome Maran Rashti from Griffith University and Richard Bell from Murdoch University here to support us and give us some information about some of the projects that they're working on in regards to soil constraints, particularly compaction and sandy soil. So Maran, I'll pass it over to you.
Thank you. I'm just trying to, I did it before, but I can do it now. Yep.
Great, that looks great.
Okay, so I'm trying to give you some update about the project that we're doing for Soil CRC regarding evaluating soil functional resilience to compaction and drought stress for developing higher performance soil. So to start with, the idea before putting this project together was that we all know that through a conventional practice that we have in our agriculture system, we're losing a lot of organic matter, and the loss of organic matter will affect the soil fertility and soil health. Environmental stresses such as drought and compaction can accelerate this process. So through our best management practices, we usually try to reduce the loss and reduce the effect on the yield that we have. But it's not easy to figure it out. What health status the soil have. So is the soil is healthy or not healthy? How we can define that? So through Soil CRC, there are different projects that are working on improving soil fertility and soil health, but there was a need for a specific tiny projects to put things together and end up with basically soil health test and understanding of the health status to be able to use in other bigger projects through Soil CRC or field experiments. So this project doesn't have field experiment. This is basically a lab experiment with a couple of incubation experiments to be able to come up with the mechanisms behind the effect of stresses on soil health and how we can measure the recovery. So to do this, we try to put a good team together. So these teams are from Griffith University. We have Professor Chen and Mariam. New South Wales DPI, we work with Professor Lucas van Zuiten and Dr. Mike Rose. And also different farming systems from the areas from North Queensland and Western Australia. The area are dealing with compaction, like the areas on Herbert, leading with Lawrence DiBella, Herbert Kent Productivity Services, and the areas that are mainly have problem with the drought, like Western Australia, FACEIT groups, led by Mrs. Sarah Hyde. About the background, we all know that a healthy soil should have the capacity to continuously supply plants with enough nutrients, water, and air to remain the complex biological systems in a healthy condition. But we know that the decline in the crop yield through extensive application of fertilizers, too many cropping systems after each other, and basically intense farming systems can reduce soil fertility and soil health, which can be extended by environmental stresses like compaction and drought that were the main focus in these projects. So in order to drive a practice change, first we need to know which parameters we're looking for. And the second thing is that parameters that we're measuring should be cost effective. So if we come up with a consortium of different parameters that are too expensive for farmers to use, so they're not going to use it so frequently, and basically the outcome of the project will be so limited. So we try to use different physical, chemical, and biological processes to find the main mechanisms through this process. And the next step through this project is to simplify it into one or two simple basically assessment or simple test that's affordable for all farmers and farming groups to be able to assess their soil health. And basically we relate it to the other parameters that we measured before. So we're trying to make a model. That model will be specific for each environmental condition, each soil condition. And then within using a simple process, for example, like measuring CO2 respiration, we can relate those results to the other parameters that we have measured in the lab and come up with the soil health status, which can be used in different field experiments to evaluate that is our best management practices are exactly increasing soil health status or not. In order to do that, we need to have a good understanding of soil resistance and soil resilience. So basically what we call a soil resistance, if a soil goes under different environmental stresses, is try to basically keep its normal condition and resist against changes. That will be different from different soils according to the texture, organic matter, and so. But if the stress happens so frequently and so commonly, the soil will affect its it will affect the productivity of the soil, whether we like it or not. And then the next factor comes into place that we call it resilience. So the resilience of the soil is the capacity of the soil to recover its main functional properties after we're removing that stress. For example, you're going through a drought season, and after that, you get the next rainfall. You want to know how fast and how much of the basic properties and essential properties of your soil will come back to its normal condition, and eventually how it will affect on your yield. And if you're using different management practices, like you're adding organic matter to the systems, how it will accelerate the recovery process. So if you look at the diagram here, when the soils goes through different environmental processes, they lose their functionality, but after removing the stress, they can recover a bit of that functionality back. But if this process happens too frequently and for a long period of time, eventually we get to a critical point that the functionality of the soil will be damaged permanently. And then we start to have soil degradation, and the recovery of the soil will be so expensive and almost impossible in the areas that in marginal land that the profit is so limited. So these projects try to put different biological, chemical, and physical properties together to have a good understanding of the mechanisms responsible for the effect of stress on soils and the mechanisms related to recovery of the soils after we remove the stresses in different farming systems. We try to understand the mechanisms and try to provide some techniques that ultimately increase the production and profitability of Australian soils. Here you can see a small device that we designed for putting compaction into our soil systems. So we know that, we're doing a small incubation experiments. So we have a small jars, we put soils in our jars, and we try to apply compaction in them and replicate the compaction that happens in our farming systems. Then we can put them in different drought condition. We keep them for 1 month or so. So we monitor the changes that happens in the physical, chemical, and biological process that happen to the soil. And we monitor to see how the soil functionality start to decrease. Then we remove the compaction or remove the stress, and then we monitor that how the soil is going to regain its functional properties back. What is the objective? We're trying to provide robust and affordable assessment methods for measuring soil functional resilience against compaction and drought stresses. We're trying to assess the effect of compaction and drought stresses on soil functional processes, microbial community composition and diversity in relation to soil fertility and plant productivity. As you know better than me, soil physical and chemical processes are more resilient to changes in environmental systems, but the main thing that makes soil alive is soil biological properties that are really sensitive to environmental stresses. As you know, we don't have a complete understanding of the different microorganisms living in soil and their functionalities, but overall, we can assess their response to the changes by basically monitoring their changes after you go through different environmental stresses. So we try to minimize, to do the different processes and then minimize the number of tests that has to be done. As you know better than me, more than 20 or 30 different tests are available worldwide for measuring soil health, but the results are not consistent, they're not affordable for farmers, and they're not applicable to every farming system. So we try to have a good understanding of the Australian process and what's happening in Australian soils and how we can minimize the number of tests that has to be done. So the aim is to provide a practical methodology using innovative biochemical methods which are affordable and fast for different farming systems to be able to understand their health status, and eventually we can basically collaborate this with other international communities and extend it to other parts of the world. What we have done, we put a team together and try to design different projects and selected different soil sites from Western Australia and North Queensland that had the problems with the drought and compaction, and we try to provide some guidelines, scientific papers and review papers regarding the effect of these stresses. Also, different technical reports and fact sheets to be used in further projects through Soil CRC, which have more field-based component, both in Queensland and Western Australia, to test the findings of these projects in action and in the field. So, progress to date, what we have done through now, we have a really good PhD students working on the projects alongside with the researchers. So we did a comprehensive literature review and we find out what parameters are mainly assessed when we're looking at the soil health in Australia and worldwide. And we try to minimize and pack them together in a way that we can handle them in different incubation experiments. After that, after we identify the main factors that people measure it worldwide, we designed our first incubation experiments, which was a 72-day incubation experiment. In this experiment, we used two soil types, a sandy soil and a clay soil under best management practice. So we didn't want the soil to have any stress at the beginning. So we basically targeted to our farming system and asked them to provide us with the best management soil that they have. Then we put these soils in different stresses, both moisture and compaction stresses. We decided to go with two moisture stresses, 15% and 35% water filled porous space. You know that especially in sandy soil with low organic matter, their water holding capacity is less than 20% of their weight. So when we're talking about 50% water filled porous space, is less than 5% moisture content in the system. So it's a pretty severe drought. And we looked at different bulk densities from 0.9 to 1.5 gram per cubic centimeter. The main aim of this experiment was to find the main indicators of resilience in the best performing soils, and we try to see the effect of compaction and drought on changes in microbial community composition and functional process. The primary result we get from this experiment was interesting. We found that the microbial metabolic activity, as we measured it through CO2 respiration and enzyme activities, responds differently in different stresses, which is varied through different soil texture as well. So if I want to explain it a bit more, when we're talking about microbial metabolic activity, it means that how microbial community react to the changes in their food supply and the water supply in the system. For example, if you have a microbial community that goes through a healthy condition with enough water and food, they increase their number. When they increase their number, they produce more CO2 because they respire CO2 the same as human does. But in this case, basically increasing in CO2 respiration is directly related to the increase in the number or the total mass. So the total mass is increasing, they're producing more CO2. And as you know better than me, they cannot basically eat foods. They need to provide enzyme for external digestion in their systems. So they basically exudate more enzymes into the systems and they respire more. But in this case, it means that they are healthy and they're increasing in their number. But what happens when they go through a stress? They may also provide more CO2 respiration. In this case, because they're suffering from stress and they're fighting for their life. It's exactly like the same time as we're running, we're doing exercise. We're not gaining weight when we're doing exercise, but we're burning more carbon because we basically need to invest more energy to survive. The same thing happens to microbial communities. Sometimes they produce more CO2, but in this case, when their body mass is shrinking, when their microbial biomass is shrinking, but they're providing more CO2, they're under stress. So when we're looking at metabolic activity, we're looking at the relationship between the amount of enzyme and CO2, the amount of enzyme they produce, the amount of CO2 they respire, related to the body mass that they have. So we want to see that if the CO2 or enzyme activity basically coupled with increasing in their body mass or coupled with decreasing in body mass, which can give us a good indication that the systems are suffering or thriving. This is a good way to find out how the systems suffer when you go through the stress and how the system thrive after you remove the stress from the system. Hope it will explain a bit. So what have we found? I just don't want to go through too much details. Just want to show you why this process is different in sandy soil and a clay soil, because sandy soils are the main soils that we're dealing in Western Australia. So if you look at the left-hand side, when we measured hot water extractable organic carbon, this method is different with measuring in cold water. So we know that in measuring total carbon, a big part of this total carbon consists of recalcitrant carbon that are not available for microbial communities. So they take a long time to be consumed as food sources for microbial community. But when we basically extract the samples with cold water, we're only looking about only dissolved organic carbon, which will consume really fast within 2-3 days. Or previous experiments shows that there is a good indication and relation between the amount of organic carbon that we measure through hot water extraction and the amount of carbon that available for microorganisms to consume. So this process is really simple. We incubate the sample with hot water for 16 hours and it will release the organic carbon in the system. So we measure it after 16 hours. So if you look at the right side, the two graphs that related to low moisture sandy soil and low moisture clay soil, which is related to drought condition, you see that when you put the soil under stress, the first 30 days of the experiment, you see their hot water extraction organic carbon decrease really fast to a really low critical point. But later on, when we remove the stress, they start to recover back. So you see that the microbial activity is increasing. You get more basically decomposition happening. You get more food source available for microorganisms. But you see the gap is different when you're talking about the sandy soil, when you're talking about the clay soil. A clay soil recover faster. And you see that there is lower amount of difference or almost no difference between different compactions. But if you have compaction in the sandy soil, the process takes longer and you get a significant gap of the recovery of the microorganisms or hot water extractable organic carbon in this case, when you have high compaction, when your soil has been under high compaction stress or low compaction stress. Another example is when we measuring the CO2 respiration, the same way as I explained to you, it's a representative of microbial activities, but we refine it according to their body mass as well to find out if they're under stress or not. You see there is a significant difference when you're looking at the clay soils and the sandy soils, and also a significant difference when you're dealing with high moisture condition and low moisture condition. In low moisture condition, after basically starting the stress at day 0, the CO2 respiration is really similar in different compaction condition, means that the compaction didn't really affect it in a dry soil. If you have a dry soil, the drought is more dominant in changing microbial activity than the compaction. But when you release the compaction after 30 days, you see that you get a different recovery process in different compaction levels. So soils do not recover the same. And you see, if you continue the experiment for another 1 month or 2, the soils has been under lower compaction stress recover faster and have a better chance to gain their functionality. But it is also different between sandy soil and clay soil. For example, if you look at a high moisture clay soil, from the beginning when you put in a stress, the low compaction conditions, there is resistance against the changes, so they can protect themselves a lot better. The same happens with the enzyme activities. For example, here I put some beta-glucosidase activities, which is an enzyme related to the carbon functionality in the soil process. You see that the stress will affect the concentration of beta-glucosidase, and the concentration increase after you release the compaction. The reason is that you get more food source available in the systems, and microorganisms start to basically produce some enzyme to digest or decompose the or recalcitrant organic matter and use it as a food source. So using this experiment, we find out, we get some preliminary understanding of the process that happens, how we can access things. Here I only show you 3-4 components. We measured over basically 40 different components. For example, here is the changes in enzyme microbial community structure. For example, you see that these soils has been under compaction and drought stresses. The first soil is a normal soil without the stress. The second one, A, and the B is the one that has been through stress. You see that the portion of different microbial community changes. And overall, for example, if you're looking at carbon use efficiency and nitrogen use efficiency, so you see that it's a soil under compaction stresses, the CO2 respiration decrease. But when you're looking at the loss of nitrogen through nitrous oxide gases, the loss of nitrogen through nitrous oxide gas increased significantly. So this process of stress, especially here, look at the compaction, not only affect the overall functionality of microorganisms, it affects the changes in their community and also the use efficiency of different nutrients and carbon in the system as well. So when we got this data, for the next step of the project, we try to divide the process into 2 different sections. One section specifically related to drought, and one section specifically related to compaction. Because here, Felicity told me that the major basically focus is on drought. So I'm explaining the next step that we're doing now regarding the drought process. So through the information we found out in the first experiment, we tried to minimize the number of parameters we used. But in this step, we try to introduce the difference between conventional management and best management practices, and also application of organic residues into the system. So for example, here we ask FACEI Group to provide us with 2 different sandy soils, 2 different farms adjacent to each other, less than like 200 meters from each other. So they collected 2 samples, one under conventional practice management for a long time, and the other one under best management practice for a long time. The soil texture and properties are really similar. The only difference between them is the practice management that happens in these 2 soils. And we also asked them to provide us with the local organic matter that they apply into the system. For example, here they use wheat stubbles. So they provide us with wheat stubbles. We try to basically put another incubation experiments to look at the effect of drought and resilience in soils to drought under best management and conventional systems, and what happens if we add organic matter to the systems, how it will affect the process. So we used 2 different drought levels in this experiment. You see we have 12 treatments on 2 different soil. The top soil is a conventional soil, the bottom one is best management soils. We use 2 levels of drought, 15% water holding capacity and 30% water holding capacity, and 1 no drought condition, which is 55% water holding capacity. So we put the soils under stress without adding organic matter at the beginning. So we're keeping the soil for 1 month, the process is undergoing. After 1 month, we're going to add organic matter to the systems and mix the soil again, put them back into the jars, and we're looking at the recovery of the soils from the water stress through rewetting process. So you can see here in the treatments, we have the control treatment with no drought and no rewetting. Then we had a different drought process, different drought treatments with application of organic matter, without application of organic matter, and we're going to go through the rewetting after 1 month, which has been a bit delayed because of the coronavirus problems. We had our labs shut down, so we have started to do this experiment, and it's undergoing now for I think 3 weeks by now. So this is from me. I'm happy to answer any questions.
Fantastic. Thank you, Maron. We do have a couple of questions in the chat box. I'm interested, we'll deal with these, with the ones that, so Peter, and feel free to unmute yourself, Peter, but I'll try and interpret your question. You had a question about measuring carbon dioxide respiration in soils. So there are some tools available for researchers or landholders to use, and one of those is Solvita, which he's indicated is perhaps not perfect and not ideal. So Maron, do you have any suggestions for alternative options for measuring carbon dioxide?
Yeah, so the things we do in this experiment is a bit different, because when you're measuring carbon dioxide in your farming system, you don't really looking at a really accurate number. You know, I don't know how to tell you. When you're doing an experiment to understand the mechanisms, you need an absolute number. Your values should be really close to accurate to be able to have a good understanding of the mechanisms. But when you're looking at the field, you want something cheap and fast. You know, when you go through the complicated processes like what we do in the labs, it becomes really expensive. So for example, I showed you the jar that we produce. So we produce a small jar, and then we incubate this jar within a 1 liter container. And that 1 liter container has a basically has a hole on the lid, which has a septum there that we can extract the gas within a syringe. So we incubate the soil for each time for a couple of hours. And then, because we don't want the system to be anaerobic, if we leave it for long, all the oxygen in the system will be consumed and there is no oxygen to be converted to CO2. So we can do these processes, for example, not more than 8 or 7 hours. So we incubate the sample in a specific temperature, because you know that change in temperature will change the respiration. So we need to control the temperature in an incubator. We keep it inside the jar, and then we do gas sampling, and then we use a gas chromatograph. So we inject the gas samples into gas chromatograph, and it will give us CO2, methane, and nitrous oxide. And that's why we can test the systems, because if the systems is on aerobic condition, you don't expect too much methane come out. So if you get too much methane coming out, means that your system is getting to anaerobic condition, it's faulty. So there are a lot of tools to check if the results are correct or not. But this is expensive.
Yes.
So I saw that systems work, the company that you mentioned, it works not that accurate, but it's really fast. It's only take 24 hours. And if you really want to basically go for a cheap option, we can modify those instruments, we can modify those processes, and we can come up with the, I don't know how to tell it, the errors of the process. If we can measure the errors. For example, you have a soil type, for the first time we can measure it in a lab with accurate response. At the same time, we do it with those cheap instruments, and we find the difference between these two processes and calculate the errors, then you're good to go to use it, the cheap version. I hope it could answer you.
Yeah, good, Miran. So, and he's wondering if that carbon dioxide respiration is influenced by microbiomass. So I guess if you're taking a measure of the microbiomass, then you can make some assumptions about carbon dioxide respiration.
Yeah, that's exactly what we do. So we need to differentiate between the CO2 that coming because of increasing in the body mass of microorganisms, or the CO2 that coming because they're under stress and they need to be more active to survive. The only way to do that is to measure their body mass. It's like that, you know, for example, I'm a heavy guy. You only see only my head, but I'm a heavy guy. Compared to other people, I respond more CO2. It's not because I'm healthy, it's because my body weight is higher, so the metabolism is higher. But if we both have the same body weight and I respire more CO2, it means that either I'm exercising or I'm under stress or I'm cold or something that's increasing my metabolism without increasing my body weight. So that means that I'm under pressure, I'm under stress, I'm under danger. So the increasing in CO2 level in this case is not mean that I'm healthy. So the only way to do that is to monitor the body mass or microbial biomass of microorganisms, at the same time the amount of enzyme that they produce. For example, if they have enough food in the systems, you don't need to produce too much enzyme to digest more, because you get your basically food really fast. But if your system is lack of food, you need to provide more enzyme to be able to get the same amount of energy that you need.
Right. Yeah. I'm imagining tiny little scales for all the microbes to sit on so that you can...
yeah, so basically what we do is a bit cruel, but we need to kill them. So we're killing them to find their mass, because we need to differentiate between the organic matter that normally we have in soil and the organic matter that we have in the cells of microbes. So what we do, we fumigate them with chloroform, and it breaks down their cell membranes. And I'm sorry to say that, but all their body parts come out. And now we're measuring the basically organic carbon without body of the microorganisms. And now we basically break them down and we measure them again, so we can find out how much coming from their body and how much has been in the soil.
Yeah. Fair enough. You've got to do what you've got to do, Mara.
Yeah.
So for the good of science, I commend you. Richard was wondering about, do the results so far suggest that microbes were quite resilient to the stresses?
It depends. For example, if you're looking at the compaction, there is a level that if you pass through that, your resilience will affect significantly. So if you're looking at, increasing compaction a bit for a short amount of time, the recovery is faster. But when we looking at high compaction like 1.5, 1.6, that sometimes we see in sugarcane systems, the process is really long. And by the end of the first experiment that we have done, because we didn't have enough samples to continue forever, but by the end of the experiment we have done, still there was a gap between the different compaction levels. And we assume that maybe we can reduce that gap by adding a bit organic matter into systems and adding a bit more food source. But overall, if there is a tiny problem that I can't basically answer Richard's question, is that both of the soils that we use in the first experiment has been under best management practice. And if in this experiment that we're doing now and we're comparing the conventional versus best management practice, we can have a better understanding of the microbial resilience in these two conditions. But over there, because the soil has been under best management practice for a long time, I may say that the microbial community were resilient, but it may give a false alarm to you because that soil has been under best management practice for a long time.
Okay, excellent. Does anyone else feel free to unmute yourself and ask a question if you like? Peter, do you want to unmute yourself to ask that question?
Sorry?
Yep. Yeah, and I was just wondering about what critical compaction level you use, because I have read that I did a project where I read and I used 2000 kilopascals using penetration resistance, but then I've recently read a measure of 3,500, but I assume what you just said then was in bulk density. Was that what you're talking about, 1.6?
Yeah, so it's a bulk density, and the maximum we went was to 1.6, yeah.
I can cover some of that in the following presentation, Peter.
Yep.
Thanks, Richard.
Okay, thanks.
Any other questions for Meron? Meron, you are welcome to stay and listen to Richard's presentation if you like, or I understand if you're busy and you need to...
No, I'm going to learn a lot from his presentation.
Yeah, great, fantastic. Okay, so if anyone thinks of any questions along the way, feel free to pop them in the chat, and if Meron has hung around, we might be able to answer them, or yeah, certainly Richard is in the house, so I'm sure he'll be able to as well. So Richard, thank you again for joining us. You've been a great supporter for the West Community of Practice, and I appreciate you making yourself available again to take us through compaction issues in the West, and if we have time, some sandy soils. So I'll leave it to you. Thanks, Richard.
Okay, let me get the screen up. So Can you see that now?
We can.
Excellent. So I did prepare something on water repellents as well, but let's just see how we go for time. I'd much prefer that we go through it slowly and deal with people's questions rather than rush through it. And look, I do apologize. I prepared it more as a lecture, and I realize that for adult learning, that's not necessarily the best approach, but I had this material, so it was easier for me to use it. But we can go through it as fast or as slow as you like. The photograph is quite telling. This is a crop that's come up in the wheat belt, and you can see these strips where the crop is quite yellow, and that corresponds with wheel tracks. So that's a really good illustration of a compaction issue in the soil. And presumably where the soil is compacted under the wheel tracks, the rainfall is not infiltrating and you're getting some temporary waterlogging, and that's what causes the crop to yellow off. It doesn't always show up quite as neatly as that, but that's a visual indication of the sort of problem that of compaction. Locally, there's quite a bit of good material that is available to read up on and learn some more. There's the soil guide, which is available from DPIRD. Jeff Moore was the chief editor of that book. It's, I guess, published or revised in 2004, so it's perhaps getting a little bit old now, but I think it's still got really good basic material in it that is relevant to Western Australia. Secondly, Steve Davies and colleagues a little bit later put out a bulletin from DPIRD on subsurface compaction, so that's a bit more up to date. Also relevant is ISBISTA 2013 on the controlled traffic farming technical manual that obviously deals a bit with subsoil compaction. And DPIRD had a publication called the Report Card in 2013 where they put together what we know about the different land degradation issues in Western Australia, where they occur, how severe they are. And there's an article in that on subsoil compaction led by Dan Carter. So what I'm going to present draws from that, those sources. So the key messages is that in the West, we do have soils that are quite susceptible to soil compaction and to pan formation. And it's been an issue around for last 30 years or more that we've been aware of. And it is important because the estimated loss of crop production is in the order of $330 million per year from that. But there are technologies that have been developed. Deep ripping's been around for quite a long time, and more recently, the inversion ploughing technologies have become popular, as well as spading. So they're engineering treatments to try and overcome compaction, and they're obviously reasonably expensive. Importantly, with the spread of controlled traffic, we now have technologies to minimize the recompaction. Because without controlled traffic, recompaction is going to occur within the space of another 2, 3, 4 years. So it's not a permanent solution, it's only a temporary solution. On clay soils, there may also be some role for gypsum in trying to improve and stabilize soil aggregation. But soil structure and compaction is a process that damages soil structure. Soil structure is very dependent on organic matter. So whatever we can do that helps to build up organic matter in the soil, retain organic matter in the soil, plays an important long-term role in resisting the effect of soil compaction, that is, making the soil resilient to that, those forces causing compaction. And a lot of what we deal with in relation to compaction is on cropping soils. But of course it is an issue in horticulture as well, and in livestock farms or in mixed farms where you've got stock movement and grazing, and particularly in the winter, that can also be a significant factor causing compaction because the soils are wet. From DPIRD, they have put out this hazard assessment for compaction, and what it shows is that in the northeast and eastern, southeastern wheat belts are the areas where large proportions of the landscape are susceptible to compaction. Other parts of the southwest, which have moderate or low hazard, it doesn't mean that compaction's not of any concern. It just means a smaller proportion of the landscape is likely to be affected as opposed to the areas that have high hazard, and that's just related to landform. When we talk about compaction, it is broadly within the context of hard layers in soils, and that's a bigger topic than just compaction, because we know that lots of soils, even under native vegetation, can be quite hard. Lots of native soils have very dense subsoils with extremely high bulk densities in the subsoil. So putting this into context of hard layers, hardness comes from packing. So just the way in which the sand, silt and clay packs together. So if you've got the right combination of sand and then silt that fits into the pores between the sand and then clay that fits in the smaller pores, then it can all bind together into a hard packing or hard setting type of condition. That can be exacerbated by transient bonding. So when the clay, sand and silt pack together, they tend to bond together. But some of that is reversible. When the soil re-wets, it softens again. And I think most of us who have worked out in West Australian wheat belt have seen soils like that, and probably also in Victoria. Soils that are rock hard in summer when they're dry, and then as soon as they wet up, they start to soften. There's also cementation. So this is where the sand, silt and clay are more permanently bonded together through compounds like iron oxides, aluminium oxides and silica. And that might be expressed in a silcrete pan, for example, which you often find in duplex soils. Just at the top layer of the duplex B horizon, you often get quite a hard layer there from silcrete that has bonded together the clay particles. So that type of permanent bonding is there regardless of whether the soil is wet or dry. So those are natural features. Also, we've got other natural processes that cause hardening of the surface. There's what we call slaking and dispersion. Slaking is a tendency of all soils when they wet up, because when you've got a dry soil and water falls on the surface and it starts to soak in, water starts to penetrate into the PEDs, and if there's any air trapped in those PEDs, as the water soaks further and further into the PED, it compresses the air that's trapped inside it, and in some non-resilient soils, eventually the PED explodes. The force of the pressure of the air that builds up within the PED is so strong that it just causes the PED to collapse completely, and that's what's called slaking. So that commonly happens on soils when they wet up with the first winter rains, and that's often expressed in a layer that's perhaps a few millimeters, 5 millimeters thick, which when it dries forms a hard crust on the surface. Dispersion is a bit different. That's related to sodicity in the soil. So those soils, when they wet, they tend to disperse because of the influence of the sodium. Raindrops, when they hit the soil surface, have quite a lot of energy, and the impact of the raindrop itself can break down soil PEDs and reduce the surface structure and lead to that crusting. Loss of organic matter by any means, usually by repeated cultivation, is a process that reduces the stability of soil structure. And repeated wetting and drying can actually be a positive influence that helps to naturally reform soil structure. Then we've got the compaction. This is where there's external load, or the weight of machinery usually, but also animals, which compresses the soil depending on their water content. And so apart from the weight of the vehicles, which usually results in subsoil traffic pans or compaction, you've got the effect of cultivation itself, which can lead to plough pans. We obviously see less of that nowadays in our wheat belt in Western Australia because of no-till. And then there's the effect of stock trampling, which causes more surface compaction rather than subsurface compaction. So that's a bit of a framework to try and put it all together under the banner of hard layers in soils. I might just skip over that one. Felicity, I'll make this available, so if anyone wants to go through it later, and or anyone who's not here today wants to look at it later, it'll be there. So this table just summarizes in text some of the things that I talked about. So how are we going so far? Are there any questions that have popped up yet?
No questions popped up yet, Richard.
Okay. Why does it matter? Here's a set of data that really illustrates why compaction layers are really significant for crops and pastures. And if you look at the table at the bottom, this is from some old data with wheat that DPIRD did for two different sites, East Chapman, which is up near Geraldton, and Wongan Hills, which is a couple of hours north east of Perth. The rate at which the roots grow through unripped and ripped soils can be very different. So in the ripped soil, the wheat roots are growing at about 14 millimetres per day. So over a centimetre, almost a centimetre and a half per day. So they're really tearing through the profile where it is ripped, but in the unripped soil, they're only growing at about a third of that rate. Which means that in the ripped soil, they can get much deeper. In the same amount of time, they can penetrate to a much greater depth. So in this experiment in the ripped soil, after 35 days, the roots were down to 60 centimetres, 50, 60 centimetres, where they can explore all of the water and the nutrients in that upper 50, 60 centimetres, as opposed to the unripped soil where they're still confined to the upper 20 or 30 centimetres. So they've just got basically half the water and nutrient resource available to them. And the figure above just shows the rate of penetration of the roots through the ripped versus the unripped layer. So it's not that roots won't penetrate through some of these compacted layers, it's just that it takes them longer, slows them down, and they may never catch up. So even later in the season, when the rain is drying up, the ones that got through that layer quickly and got their roots down much deeper are just able to tap into subsoil moisture reserves, keep them going a little bit later. And we know 1 millimetre extra rainfall from the subsoil late in the season could be worth 40 or 50 kilograms extra grain per hectare. So a few extra millimetres of rainfall has a really noticeable effect on the bottom line. And these are some yield increases from experiments done by DPIRD over the years. Some old work that Bill Crabtree did in the 1980s, and then more recent work of Steve Davies or Ron Jarvis and then Steve Davies. And you can see these are quite impressive improvements in yield, 1/2 tonne or more. And they're data sets that are based on lots of comparisons. So there's pretty robust data on loamy sands and other sands where you can get quite significant boosts in yield with a deep ripping treatment on soils that have subsoil compaction.
And Richard, was that was the depth of ripping, was that specific to each soil compaction depth? So, for example, yeah, were they ripped at the depth where the compaction depth was identified, or were they all just ripped at like 20 centimetres or 30 centimetres, or, you know?
Look, can't answer that specifically, but the general guideline is that you would identify where the compaction layer is and make sure that you rip about 50% deeper than that. But out of 50 or 60 trials, couldn't be sure that everyone did that diagnosis first before setting up the trial, or whether they, you know, some did and some didn't. People like Ron Jarvis and Steve Davies are pretty systematic and methodical. I'm sure a lot of those trials would have been done by firstly digging a auger hole or a pit, identifying where that compaction layer is, and making sure that the depth of ripping exceeded the depth of compaction. And of course, you need to do it at the right moisture as well. So if the soil is too moist, then you don't get the same fracturing and shattering and breaking up of that layer. If the soil is too wet, it can be just like, the ripper can be like a hot knife through butter. So it does make a impression in the compacted layer, but only very localized. Yeah.
Is there a guide for moisture levels for different soil types for ripping anywhere?
Oh, look, in our climate, you've got a long period when the soil is dry, and you're most likely to get effective ripping on the sands. Of course, if you're dealing with clays, it might be a little bit different, where you might have to be a bit opportunistic, and if you get some summer rainfall and subsoil moisture, ripping after that might be easier to penetrate, but still sufficiently dry to get that fracturing and cracking happening. What's your experience over in Victoria?
Yeah, I probably don't have enough knowledge to share in that respect, but certainly I went to a field day early in the year, probably in end of February, March. It was pretty, we had a bit of rainfall during February, and although it was pretty dry, and they were doing a deep ripping demonstration actually, and it was coming up pretty cloddy. And it was pretty dusty as well. So yeah, similar to you, Richard, that balance between of getting a bit of moisture so that you don't end, so that you can, yes, get the shattering, but not so wet that you get that shearing. So
yeah, look, Western Australia, we've probably done most ripping on sands or loamy sands, but there is interest in ripping on clays. And Nadine, are you still there? I think up in your area, they've been doing a bit of ripping in clay soils. Do you want to add anything to that?
Hi guys. Yeah, they haven't had great results, and it has been very cloddy, the paddocks I've seen. And I know that Steve Davies is not super confident about it. It's sort of the trials that he's done don't always show up real well either. So it's probably a risky move, but I think people are desperate for ideas, and that's why they try it. But Steve Davies would know obviously a lot more than myself.
Yeah, that's my understanding as well, that it's an area that we'll probably see a bit more work over coming years to see if the research can work out when it will be effective and cost effective, and when you should avoid it. And obviously a lot of the clay soils have sodic subsoils in the southern wheat belt, also high boron, salinity. So those are issues that you'd have to be pretty cautious about, and before a big investment, you'd need to be confident that it was going to leave you in a better shape than you were before. Shall I continue?
Yes, thank you, Richard.
Good. I did refer to soil structure before, and we're all familiar with soil texture, which is the percentages of sand, silt, and clay. So those are what we call the primary soil particles, and that has a major influence on pore size distribution within the soil. So the proportion of macropores that allow roots to grow through, air to penetrate, and drainage, as opposed to the mesopores and micropores, which are the finer pores that will hold water and store water. But soil structure is where the sand, silt, and clay get reorganized into secondary particles, which we sometimes call PEDs or aggregates. And the extent to which that happens in the soil determines how well structured the soil is. Now, we have a lot of sands or sandy A horizons in Western Australia, and sandy soils don't form good soil structure. We do have in duplex soils and soils with clay horizons some structure formation, but when we teach undergraduates in Western Australia, it's actually quite a difficult concept to teach, because we just don't have a lot of soils with really well-formed structure. Not like if you go to the Darling Downs of Queensland, or some of the well-structured soils in Eastern Australia, or some of the super well-structured tropical soils that I've seen. But the PEDs can have different shapes. The most common one in the A horizon would be this crumb structure or granular structure, and that's largely due to the influence of organic matter and clay. In the subsoil, particularly in duplex soils, you often get this columnar structure or prismatic structures. But other structure or PED types that you might see are angular or polyhedral, and platy type structures are something to look for, as they often occur as a symptom of smearing or plough pans, and they may be evidence of compaction. So how do PEDs or aggregates form? Well, it's where sand grains and clay domains and organic matter, together with polyvalent cations, so particularly calcium and aluminium, chemically they just bind together to form this secondary structure. And when you look at the soil profile and you dig it up with a spade, you'll often see these PEDs just sort of fall out of the soil material. They have enough binding and coherence together to form those PEDs, and they change the pore size distribution of the soil. So on sands, they create more mesopores and micropores, so you get greater water holding. And on clays, they tend to break it up so that you get more macropores that allow better aeration and drainage and root penetration. So any questions on that?
Now, Nadine's just asked, is there a simple farmer-friendly guidelines on what tests to check before ripping and what the thresholds are, especially for clay or loam soils that are more risky? yeah, I don't know if you just want to make a quick comment, or not a quick comment, but a comment about that.
Look, off the top of my head, I don't have an answer to that, but I can do a little bit of searching and see if I can come up with something.
Yeah, these are the sort of things, I guess, Nadine, that we were wanting to do out in the field, and when we could get a soil pit. But yeah, I mean, off the top of my head, thinking about things like those constraints, salinity, city, boron, those sort of things that yeah, Richard already mentioned, and I guess getting some thresholds. So considering what crop you might be growing next, or what your crops you're planning on growing in the future, and their tolerance to those sort of levels.
Yeah.
Yeah, we can see if we can find some.
Yeah, look, I think you'd probably assume if you're ripping a clay soil, you're going to bring up some clods from the subsoil. So you would want to be sure that there's nothing particularly nasty down there.
Yeah.
Because if you bring that up to the surface, then you're just exacerbating the problem.
Yeah. Thanks.
Yeah, it'd be really good to, I guess, see a bit of a culture where people do check more. I think that sometimes, we'll just do a bit of ripping and see what happens. So it'd be great to encourage more checking before digging somehow.
Right, no checky, no learning.
That's right.
Yeah, no, I think that's a good mantra to keep repeating. You can't manage what you haven't measured, and digging a hole with an auger, with a spade, can tell you a lot about a soil profile.
Yeah, okay. So it doesn't have to be a fancy test, just to dig with a shovel is sometimes a good idea.
There's a lot you can see just from digging a hole and looking at the profile. If you go down to 50 centimetres or so, you can pick up a lot of the significant aspects of the soil profile. But once you've done that, you may as well take a soil sample and get it analysed as well.
Yeah.
Now, I've just got some workmen who've come to the front door. I just need to let them in. So can I take 2 minutes break?
Yes.
Chat amongst yourselves.
We'll chat amongst ourselves. No worries. Thanks, Richard. Yeah, I'm really big on that. You can't manage what you haven't measured. And if you had, like Richard said, I mean, there's some field things that you can do in the field. So even if you had a pH test kit that you could, you just get them from Bunnings. So if you're looking below that, like at any incremental stage, you could do some pH, so you can have a look at whether it's increasing or decreasing to determine if you've got a pH constraint. You could also do even the slaking and dispersion tests at depth. So again, taking some little pads of soil and using rainwater and just popping them in. And I'll put some video links when I send the videos. The link to this video, I'll include some YouTube videos so you can have a look at the slaking and dispersion test. But that will just give you an indication of any sodic issues at depth. I think salinity is probably an issue that would be for you guys. So, but that's a lab test. I don't know of an in-field test for salinity, but if anybody else does, feel free to pipe up. So they would be, a few tests. Boron, I'm not sure of the boron issues that you guys might have over there, but we've certainly got some boron at depth in some of our clay soils in Victoria. So, yeah, I think they're just a couple of things, even just a texture test to give you an idea of what the texture is like at depth. So if it's getting sandier or if it's getting clayier. And Peter, there could be a risk of bringing up acidity from ripping. Although, Richard, you might have a more experience in that. Is there a risk of bringing up acidity with ripping sandy soils, or is it mostly clays?
No, certainly on the sands, a lot of those have acid subsoils. So a lot of farmers nowadays, when they rip, are also liming. and using that as an opportunity to try and incorporate lime and get it down into the subsoil. And it might be combined with spading, moldboard ploughing, one-way ploughing, just to get that incorporation of the lime while you're doing the ripping operation.
Lucy, I wanted to add that I remember going to a workshop and that was just all about digging holes out in paddocks, and one of the things they always recommended was dig a hole under a fence line, because that's where there isn't any traffic and you get a comparison of what the soil may have been like before it had chemicals and fertilizers and high traffic type situations, and even, you know, you don't get the same kind of stock traffic under the fence line, so it makes a good comparison with where you are in your paddock versus to what it might have been prior to that.
Yeah, great, Belinda. Good, very excellent point. Yeah, thanks.
Yeah, there's a few folk in DPIRD in Western Australia who are really good on this field diagnostics with pits, backhoe pits and the like. Jeremy Lemon, who's based in Albany. I've seen Jeremy in the pit, and you know, he's just really experienced and really good at being able to go through the things to look for. And Steve Davies up at Geraldton also is really good in that sort of setting. So if you can get either of those people along, you get good value out of them.
Yeah, great. Thanks.
Yeah, our last 2 field days we had some soil pits and it was really good, but probably need to keep doing it, I think.
I think so.
Too many soil pits.
Yeah. I went to on a field trip years ago to a vineyard near Ballarat, I think it was, and they just talked about how this vineyard had been established for over 20 years, and try as they may, the vines just struggled and hardly produced any fruit. And they tried everything until they invited a soil scientist along. He said, can we get a backhoe and dig a pit and have a look at the profile? And what they found was that all the roots were in the top 30 centimetres, and below 30 centimetres was just a really dense clay subsoil. They put a ripper through that. And after that, the vines just took off and thrived. So, you know, you can learn a lot from digging that hole, and you pick up the basic constraints. There may be some other things that are a bit more tricky to diagnose, or where you need to do a soil test as well, but don't underestimate the value of that soil pit or looking underneath your feet. Shall I continue?
Yes, thanks, Richard.
Okay, just a little bit more about soil structure. Roots themselves, as they grow through, they do help to create soil structure. Firstly, by the pressure that they create that pushes particles together, as well as the organic matter that, or organic compounds that roots excrete. All of that helps to create a biological environment for building soil structure. The key with soil structure, apart from the clay, is the organic matter and its activity in the soil. And it's not just a matter of having the organic matter there, it needs to be turning over and producing gels and gums that microbes produce, and that helps with the binding together of the clay, sand and the cations. I mentioned wetting and drying. Wetting can have a negative effect on structure where you get slaking, but it also has a positive effect on structure, because as the soil dries, the particles actually rearrange and come closer together, which increases the bonding between the sand, silt and clay and organic matter. That happens more with some soils than others, and some clay soils are quite self-repairing. As they dry, the structure reforms. We don't have a lot of those sort of soils in Western Australia, but in Eastern Australia they do. Not so relevant in our environment, but freezing and thawing is a process that can help reform soil structure. And tillage, we think mostly of the negative effects of tillage on soil structure, but you can get some, if you till soils when they're not too wet or too dry, you can actually get some beneficial influence on forming soil structure. And the photograph there on the right just shows a clay subsoil, and you can see the way in which it's cracked and fractured, and that's the indication of the PEDs, which in that particular soil are reasonably large PEDs. So the importance of soil structure and maintaining the soil structures around drainage, having enough macro pores so that water will actually drain, excess water will drain through the profile. Secondly, you get storage of water in the PEDs for dry periods. Thirdly, that there's enough macro pores that air can penetrate through the soil and provide the aeration for roots and microorganisms. And finally, for root growth itself. You have to remember that roots are delicate biological structures. They're not like a metal rod that just sort of jams its way through the soil. They actually have to find pores that are about the same diameter as the tip of the root to actually grow into the pore. So if you don't have enough of those macro pores, it's difficult for the roots to actually penetrate through those layers. Although in Western Australia, what we often find in these soils that have a dense V horizon, that the roots will tend to penetrate through old root channels. And these old root channels might have existed from the native vegetation. They may have partly filled in, but they remain sufficiently open that successive generations of crop and pasture roots will regrow through those same channels. Here's an old table from a standard soil physics textbook, just showing how the plant available soil water contents can be improved if you've got good structure. So regardless of the texture, if there's good soil structure with plenty of PEDs, you can increase the amount of plant available water quite substantially. The things that I'm going to focus mostly now on subsoil compaction. And subsoil compaction comes from three main processes: compression, shearing, and smearing. So, and compression is probably the main one that we now focus on, particularly in the broadacre agriculture. And that's just the weight of machinery and animals causing a vertical force, which is transmitted into the soil and causes the particles to settle closer together. But if you've got wheel tracks and spinning or slipping of the wheels or the tires of vehicles, that can cause shearing, which is another form of compaction. Smearing, particularly when we did a lot of tillage, the tillage tines, or if it's rotary tillage, the force of those causes smearing of the soil. And this photograph on the right-hand side is actually one from Bangladesh, where we're using really small machines. And this is what's called a strip tillage, where you've got the rotating blades that have been pulled through that soil to try and open up a slot to place the seed and fertilizer. And you can see the soil was just far too wet, and it's just caused that smearing along the side. And once the clay on that smeared layer starts to dry, it sets rock hard. And what we were finding is that the seeds that we were placing in that slot, the roots just weren't able to penetrate out of that smeared zone. It was so hard and impenetrable that the crop grew very, very poorly. In the subsoil or the subsurface, there's two main types of compaction. One is the plough pans and the other is traffic pans. So the plough pan is where you've got repeated tillage at the same depth. And that causes smearing towards the base of where that tillage layer is, that over year after year, just forms this quite dense layer. Often there's quite distinct boundaries between the upper and lower part of that smeared layer, so that when you look at the profile, you can often see that as quite a distinct layer. Now that would have been very, very common in our wheat belt 30 years ago, when everyone was ploughing 2 or 3 times a year to establish crops with no-till. It's not such a big issue, but even with no-till, if you're going in when the soil is too wet, the knife points and the other points can lead to a degree of smearing. But there are many other forms of agriculture and horticulture that still do ploughing. So the plough pans there are issues that you need to be familiar with and aware of. Then the traffic pans, these mostly come from just the compression of the weight of machinery that leads to a layer of very high strength, normally 10 to 40 centimetres into the soil profile. And that is more common in sandy and loamy soils than it is in other textured soils. This is an indication of the different pressures that animals and machinery can cause. And you can see that horses and cows, under their hooves, can actually create the same amount of pressure that a large tractor does. With sheep and humans and small tractors, the pressure is not quite as much. And the compaction in these layers is very much influenced not just by weight, but also by the number of passes. And it's a cumulative sort of thing. One pass, you get a degree of compaction. 5 passes, you can see that the compaction is spreading deeper. And 15 passes, you can see that it's spread quite deep into the profile, 75, 80 centimetres or more. And even the very compacted layer might still be going down to 30 or 40 centimetres. And you can often see those compaction layers. So here's a photograph from one of the DPIRD publications, I think taken by Steve Davies. And in that photograph, you can actually see this very distinct compaction layer in a soil that is probably a sandy loam texture. So in that layer, you've got a decrease in pore size, volume, and also decrease in pore continuity. So it's difficult for roots to penetrate, difficult for roots to access water and nutrients in that layer or below it. And you may also get increase in some of the harmful pathogens as a result of those compaction layers. This is from one of the DPIRD publications, which indicate these subsoil compaction layers probably form more in the loamy sand and sandy loam, sandy clay loam textures than other textures. How do you measure it? Don't underestimate field observations. That photograph I showed earlier, you can often see that as plain as day when you dig a hole. and you don't need anything more fancy than the hole to show that there's a problem. Another way is penetration resistance using a penetrometer, such as shown in the photograph here. Do need to be aware that the penetration resistance is influenced by moisture. So you should wet up the soil, let it drain for 24 or 48 hours first before taking the measurement. Otherwise, dry soils will always have much higher penetration resistance than wet soils. And if you've got variation in moisture from site to site, or measurements taken in summer versus winter, then it will be largely due to the moisture rather than due to the inherent compaction within the soil itself. And then the other one is soil bulk density, which is taken with a core of known volume, which you then weigh to determine how much soil is in that volume. Seems really easy, but actually there's a bit of technique involved in collecting good soil bulk density data. Now, how are we going for time? Because
yeah, thanks, Richard. I was just going to say that we have hit our hour and a half time. So I'm not sure if you would like to sort of wrap up. I don't have any other questions listed. So yeah, I'm not sure if you want to take 5 minutes to sort of wrap up or.
Yeah, look, thank you. Question was asked earlier about with penetration resistance, what's the magic number? So here's an example of a penetration resistance in a profile. On the right-hand side is the unripped soil. And you can see that at 20 centimetres or so, the penetration resistance goes above 3 megapascals. And where it's ripped, that has reduced the penetration resistance at that 20 centimetre layer to less than half a megapascal. Basically at 3 megapascals, roots will stop growing. At about 1.7 megapascals, they will definitely slow down. And we often use 2 as a threshold. So if you've got subsoils with penetration resistance above 2, you really need to act on it because it's going to be having significant effect on roots. The other question was what sort of bulk densities are problematic, and it does vary a bit with texture. So in a sand, anything above 1.65 is restricting growth, above 1.85 will stop growth. In the case of clay loam soils, they require lower bulk density in order to get good root penetration. So look, I think I might leave it there. There's a few other slides. Controlled traffic is obviously a great breakthrough in terms of maximising the value of deep ripping, because otherwise within 2 or 3 years, soils will recompact. In Western Australia, I think currently we reckon 25–30% of farmers are in controlled traffic. There are parts of Central Highlands of Queensland where I think it's probably 70 or 80% of farmers are into controlled traffic, but I think that's rapidly changing and would be more in the northern region where Nadine works than in the southern wheat belt. And that's about trying to get the percentage of the soil that is actually compacted by traffic down to about 10% compared to random trafficking, where it might be more than 40% of the soil in one season is getting the effect of compaction. So look, I'm going to wrap up there. There's a Jeremy Lemon looking at a backhoe pit and taking people through the diagnosis of pH, compaction, where the roots are, to try and work out what's the problem in this particular field that the farmer needs to focus on in order to get the best return from investment in overcoming soil constraints. So thanks for your time, thanks for listening, and sorry we didn't get quite all the way through that, but hopefully that's something useful for you.
Fantastic. Thank you, Richard. That's a great introduction, and certainly, you know, has raised a few questions and some more information that we can gather for the communities of practice. I was thinking, and I just wrote in the chat, that the soilquality.org.au website may have some resources and thresholds and those sort of things. Do you think, Richard?
Yes, look, I should have mentioned that. There's already 4 of these e-books on soil quality that have been released, and there'll be a series of others, and they are a really good contemporary resource that gives quite up-to-date information on a whole range of aspects of soil management and soil quality in Western Australia. So you can access that through the soilquality.org website. And if you've got a Apple iPad or laptop, you can download that onto your device.
Great. I'll when I send this recording of this, I'll include the soil quality website as well, which I think I've sent before, but never hurts to have it multiple times. So thanks again, Richard. I really appreciate you spending the time with us again. And to Maron, thank you very much again for joining us from Griffith University. We'll look forward to following your research. And I did, in the chat, at the top of the chat box, I put a link to Maron's project, and there's a little YouTube video that explains a little bit more about the project as well. So I'd encourage you to go to the Soil CRC YouTube channel and have a look at some of those videos that introduce each of the projects. So thank you all again. Thanks very much for everyone for participating. And
Thank you for your time.
Questions, please just post them or email. And if you've got any questions afterwards, I'll hang around to the end of the meeting if you want to have a chat about anything. So thanks very much again, everybody, and I look forward to seeing you all soon. Stay well and stay safe.
Okay, thanks all. Great to see you. Bye.